I'm his boy. This is Luke and Diego, and this is our cell signaling transduction pathway project. We're doing insulin. Our receptor for this transduction pathway is insulin, which is also a ligand. And insulin is helpful to cells because it increases tissue development and growth and maintains glucose homeostasis. It occurs in many forms, often occurring from glucose concentration building up in the bloodstream, which subsequently increases the levels of insulin. Insulin binds on the receptors called the insulin receptor, which is a receptor that consists of two identical structures. Each one of the chains consists of two subunits, the alpha and the beta, which are attached by disulfide bridges. The two alpha units on the outside of the cell create the pocket or unit where insulin binds. The beta subunits span the membrane and into the cellulose and contain a section of tyrosine kinase proteins. Insulin is the primary receptor or ligand and binds to the two receptors of the alpha sections. The two alpha sections seal around the protein, causing the beta receptor tyrosine kinases to move together also, forming a dimer. The dimerization activates the tyrosine kinase region and allows phosphorylation of tyrosine receptors by the tyrosine kinases of the opposite monomer. The phosphorylation occurs due to ATP turning into ADP. With this process, the signal is transduced from outside the cell to into the cell. The activated tyrosines act as binding points for IRS proteins, which are insulin receptor substrates. IRS phosphorylated by the insulin receptor kinase becomes active. IRS protein functions as an adaptive protein. Instead of activating anything, they act as an attachment points for other proteins. The IRS, in this case, acts as an attachment point for a lipid kinase called phosphoinositide 3 kinase, or lipid kinase.
The lipid kinase 3 contains a region that attaches onto the phosphorylated residue of the IRS molecule. This causes the active site of the lipid kinase to move into the area close to the phospholipid membrane. When the active site moves there, it causes the phosphorylation of a specific type of fat molecule known as PIP2. PIP2 has a polar head and nonpolar tails in accordance to the affinities of the membrane. The lipid kinase then places a phosphoryl group onto the PIP2, creating PIP3. The PIP3 moves in the membrane until binding onto PIP3 dependent protein kinase. When binding to the PIP3 dependent protein kinase, also known as PDK1, the protein is activated. Once PDK1 is activated, it goes on to activate the inactive form of AKT, also called protein kinase B. Once it is activated, AKT is not membrane bound, being able to move anywhere in the cell. The AKT can then be responsible for the functions of activating enzymes that transform glucose into glycogen and also stimulate protein transporters which move into the membrane and cause the reabsorption of glucose in the cell. This allows for cells in muscles, fat, and liver to absorb glucose that is in the blood to be used as energy. Thank you, Ms. Boyer.